Okay, let's talk about vision. So vision is our response to light, and we detect light by the presence of receptors called photoreceptors. So the photoreceptors, bad marker, photoreceptors that we have, they are found in our retina, okay? And there are two types there. You have your rods and your cones. Structurally, they all look alike, so more or less. So we're going to draw for you a typical structure of a photoreceptor. Let's draw one, maybe a rod, for example, like that. Okay. So this outer part here has these things called membranous discs. These are discs. And these discs contain on them these little things that we call photopigments. And it is these are the pigments that respond to light. So light acts on the photopigments. Structurally, the photopigments have just looked up. So photopigments have an outer protein part called opsin. Oh God, boy's wet. Lost that marker. Called opsin. And a vitamin A derived part called retinol. And usually at rest, that retinol is what's called a cis form. So you have cis retinol linked to opsin. That's how you form your photopigments. So this is what happens. When light strikes it, so this is light, when light strikes the photopigment, it converts the cis retinol into trans retinol. So now you have this instead. Here's the opsin part, and now the retinol is like this. And it's transformed. Okay? Now at some point, these two things will dissociate into the trans retinol separated from the opsin. And then these two will reassociate back. Reassociate back in the pigmented epithelium of the retina. And so then it can be used again. But each time light hits it, it turns into this form, breaks apart, and must be recoupled before it can be used again. So when it's in this form, the transform, this also creates some changes which allows for us to see things or not see things. So let's look at what happens once it's in the transform. Okay, I have a small board, so I'm going to erase again and start here in the transform. So imagine this is your cell membrane. These are on the photoreceptors. So here's a membrane hanging out here. Again, here is the opsin with the cis configuration. Okay. Light strikes it. It turns into opsin that's now in the transform. Okay. And when that happens, this trans opsin structure will activate a G protein. That G protein is what we call transducin. Okay. So then G protein then from there will in turn activate another enzyme in the membrane called phosphodiesterase, PDE. And once G protein activates phosphodiesterase, this enzyme in turn, its job is to convert cyclic GMP, convert it into regular GMP. So light strikes the photopigment, converts cis to trans retinol, that activates the photopigment in such a way that it will activate a G protein called transducin. Then that G protein will in turn turn on an enzyme called phosphodiesterase. Then that enzyme will convert a molecule called cyclic GMP, cGMP, into regular GMP. And that has consequences because this is what happens. So in the cell membrane as well, you have gates, ion gates. Okay. And these ion gates will let in sodium and calcium. Okay. And the gates are held open only when cGMP is bound to it. So it requires cGMP must be attached to the gate to open that ligand-gated channel. And so when you have a situation where the cGMP is being converted into just GMP. So here it was cGMP. Now it's been converted to GMP by your phosphodiesterase. That will close the gate. And so these ions cannot come in anymore. So whenever there's light, the gate closes. And that has consequences. So let's show you what the consequences are again. So we have again a short screen, small table. Let's erase it and walk some more. So here we go. In the retina, in the retina, you have three cellular connections. So in the retina, you have your photopigments up top here, which are synapsin with bipolar cells, which synapse with ganglion cells. So photopigments synapse with bipolar cells, which synapse with ganglion cells. Okay. Those are the three cellular connections inside the retina. So let's begin this process by discussing what happens in the light. So it's dark. So when it's dark outside, when it's dark to your eyes, when the lights are off, for example, here, the gates, remember your gates from the earlier screen, where you have your cGMP. cGMP is there, attached to the gate, and that allows for sodium and calcium to enter the photoreceptor. Okay, and that will depolarize it. Okay, depolarize the cell such that it will then release a neurotransmitter onto the bipolar cell. So this is an inhibitory NT that's being released from the photoreceptors onto the bipolar cell. And because it's now inhibiting the bipolar cell cannot activate the ganglion cells. So there's no action potential in the optic nerve, which is what your ganglion cells form, right? So there's no action potential there, and so there's no vision here, no vision in the dark. And by the way, this incoming flux of ions is called the dark current. Refers to the movement of sodium and calcium into the photoreceptors in the dark because the gate is being held open by cyclic GMP. Now, in the light, something else happens. So when there's light going on now, so here you have, again, the photoreceptors are here, attached to bipolar cells, attached to ganglion cells, okay? So now, in the light, remember that light will cause your cGMP to be converted into GMP by the enzyme phosphodiesterase. It does that. When that happens, that closes this gate. So now there's no dark current in the light. And because there's no dark current, there's no NT being released from the photoreceptor onto the bipolar cell. So now the bipolar cell is disinhibited and therefore able to release an excitatory neurotransmitter that will generate action potentials in the optic nerve to allow for vision. And this is why you see in the light and not in the dark. Okay, we're going to pause there.